The cell now has its genome, the information system that tells it what proteins to make, how to respond to signals, and what kind of cell it is supposed to be. But before any of that information can be used, the cell has to create something more basic, an inside that is physically and chemically different from the world outside. That is the job of the plasma membrane. Calling the membrane a border is useful, but it can also be misleading because the membrane is not a solid wall that simply seals the cell shut. The cell still has to take in nutrients, remove waste, receive signals, move ions, change its shape, form vesicles, and eventually divide. So the membrane has to perform two opposite jobs at the same time. It must separate the cell from its environment while still allowing the cell to communicate with that environment. It must be stable enough to preserve the internal conditions of the cell, but flexible enough to support transport, signaling, movement, secretion, and division. At the center of this design is the phospholipid bilayer.
A phospholipid is an amphipathic molecule, which means that its two parts behave very differently in water. Its head is hydrophilic and interacts comfortably with water, while its tails are hydrophobic and try to avoid it. Because there is water on both sides of the plasma membrane, the extracellular fluid outside and the cytosol inside, the phospholipids arrange themselves almost automatically. Their hydrophilic heads face the water on either side, while their hydrophobic tails turn inward and hide together in the middle. The result is a double layer of phospholipids, a bilayer with water-facing surfaces and an oily hydrophobic core. That hydrophobic core gives the membrane its basic barrier function. Small nonpolar molecules, such as oxygen and carbon dioxide, can pass through it relatively easily. But ions and most polar water-soluble molecules cannot simply cross the lipid interior. They need membrane proteins to provide a controlled route across. We will examine those transport mechanisms in the next board, but for now, the central principle is simple. The lipids create the barrier, while the proteins determine how that barrier is used. This is why the plasma membrane is described as a fluid mosaic. It is fluid because the phospholipids are not permanently fixed in one position. They move laterally within their own leaflet, and many membrane proteins can also move within the plane of the membrane. That mobility allows the membrane to bend, form vesicles, fuse with other membranes, extend at the leading edge of a moving cell, and reorganize when the cell divides. A completely rigid membrane might preserve a boundary, but it could not support the dynamic functions required for cellular life. The membrane is also described as a mosaic because many different proteins are embedded within it or attached to one of its surfaces. These include receptors that detect extracellular signals, channels that create selective pores, carriers that move specific molecules, pumps that establish ionic gradients, adhesion molecules that attach the cell to its neighbors or to the extracellular matrix, and enzymes that perform reactions at the membrane surface. The proteins are therefore not simply suspended decorations within the lipid bilayer. They are the working machinery of the membrane. Broadly, membrane proteins can be divided into integral and peripheral proteins. Integral membrane proteins are embedded within the bilayer, and many of them span the membrane completely, with one part exposed to the extracellular environment and another facing the cytoplasm. This makes them particularly suited for communication and transport between the two sides. A receptor can bind a signal outside the cell and transmit a change to the inside, while a channel or transporter can provide a selective path through the hydrophobic core. Peripheral membrane proteins do not cross the bilayer.
Instead, they attach more loosely to one surface, where they often connect the membrane to intracellular signaling complexes or to the cytoskeleton underneath it. So some proteins cross the border, while others organize the machinery along one face of it. Cholesterol is another important component of the membrane because it helps regulate membrane fluidity. It is important not to describe cholesterol as simply making the membrane more fluid or more rigid, because its effect depends on the conditions. When the membrane becomes excessively fluid, cholesterol restrains the movement of the phospholipids. When the temperature falls and the phospholipids begin to pack too tightly, cholesterol prevents them from locking together completely. In this way, cholesterol acts as a fluidity buffer, keeping the membrane flexible enough to bend and reorganize, but stable enough to preserve its structure. The membrane is not organized uniformly everywhere. Certain regions are especially rich in cholesterol and sphingolipids, forming small dynamic domains called lipid rafts. These rafts help gather selected receptors, adapter proteins, and signaling enzymes into the same local area. That matters because signaling depends not only on whether the correct molecules are present, but also on whether they are close enough to interact efficiently. A lipid raft, therefore, acts as an organizing platform, bringing the components of a signaling pathway into the same small neighborhood. Now we come to the most important idea on this board. In a simplified diagram, the two leaflets of the bilayer may appear identical, but biologically they are not. The outer leaflet and the inner leaflet have different lipid compositions because they face two different environments and perform different functions. The outer leaflet contains large amounts of phosphatidylcholine and sphingomyelin. It also contains glycolipids and glycoproteins, whose carbohydrate chains project into the extracellular space. Together, these carbohydrates form part of the glycocalyx, a sugar-rich coat that protects the cell surface and contributes to cell recognition, adhesion, and communication with the surrounding environment. The inner leaflet has a different composition and is enriched in phosphatidylethanolamine, phosphatidylinositol, and phosphatidylserine. Phosphatidylserine, which we will call PS, is especially important because its location carries biological meaning. In a healthy cell, PS is actively kept on the inner leaflet of the plasma membrane by ATP-dependent enzymes called flipases. This means that membrane asymmetry is not a passive arrangement that the cell establishes once and then forgets. The cell continuously spends energy to maintain it. That immediately raises an important question. Why would the cell spend ATP simply to keep one lipid hidden on the inside? The answer is that PS has one meaning when it remains inside the membrane and a completely different meaning when it appears on the outer surface. On the inner leaflet, PS is part of the normal membrane architecture. When it becomes exposed on the outside, however, it acts as a signal to the surrounding cells. During apoptosis, the orderly, programmed death of a cell, the normal distribution of membrane phospholipids changes. Flipase activity is reduced, other enzymes redistribute the lipids, and PS moves to the outer leaflet. Macrophages and neighboring phagocytic cells recognize the exposed PS as an eat me signal. The message is simple. This cell is dying and it should be removed. Because the cell is engulfed while its plasma membrane is still intact, its internal contents do not spill freely into the surrounding tissue. This is one of the reasons apoptosis is usually a quiet form of cell death that produces little or no inflammation. We will return to the complete mechanism of apoptosis later, but the essential seed is planted here. A healthy cell keeps PS hidden, while a dying cell displays it. Severe energy failure can also disturb membrane asymmetry because the cell may no longer have enough ATP to maintain the activity of its flipases. The membrane may still appear intact for a period of time, but its internal organization is already beginning to fail. This illustrates a principle that will return throughout cell injury. The cell often loses control of its functions before it loses its structure completely. The plasma membrane is therefore much more than a lipid envelope. It is an active, organized, energy-dependent system whose lipids are distributed deliberately, whose proteins control transport and signaling, whose cholesterol stabilizes fluidity, and whose specialized domains organize molecular interactions. A useful way to picture it is as a living border.
The phospholipids create the flexible boundary. The integral proteins form its gates and sensors. The peripheral proteins organize the machinery along its surfaces. Cholesterol keeps the border within the correct range of fluidity, and lipid rafts bring signaling teams together. Phosphatidylserine is the card that the healthy cell keeps face down on the inner surface. The cell spends energy to keep that card hidden, because when it turns outward, the meaning changes completely. This cell must be cleared. The one sentence to keep. The plasma membrane is a fluid phospholipid bilayer whose proteins control transport, signaling, and adhesion, whose fluidity is stabilized by cholesterol and organized into lipid rafts, and whose two leaflets are deliberately asymmetric. Most importantly, phosphatidylserine is actively kept on the inner leaflet by ATP-dependent flipases, while its appearance on the outer surface signals that the cell is dying and should be cleared. The membrane has now created a protected internal environment, but that protection creates the next problem. The same hydrophobic core that keeps the cell intact also blocks ions, glucose, and many other molecules that the cell needs. So, how does anything actually cross? That is the logic of membrane transport. The plasma membrane has solved one problem by creating a protected internal environment, but in doing so, it has created another. The hydrophobic core that keeps the cell intact also prevents many essential substances from crossing freely. The cell needs oxygen, glucose, amino acids, and ions, and it must continuously remove metabolic waste. So the question is no longer whether the membrane should allow movement, but how it can allow that movement without losing control of the boundary it has created. The first step is to ask which molecules can cross the lipid bilayer on their own. Small, non-polar molecules, such as oxygen and carbon dioxide, dissolve in the hydrophobic interior of the membrane and move directly through it. This is simple diffusion. The molecule moves from an area where it is more concentrated to an area where it is less concentrated, and no membrane protein or direct energy input is required. Ions and most polar molecules behave very differently. Because they carry charge or interact strongly with water, entering the oily center of the bilayer is energetically unfavorable. They therefore require protein assistance. Once we understand that distinction, membrane transport can be organized around one central question. Is the substance moving down its gradient, or is the cell trying to move it against that gradient? That is the real difference between passive and active transport. In passive transport, a substance moves down its gradient, from a state of higher potential toward a state of lower potential. The cell does not need to spend metabolic energy to drive that movement, because the gradient itself provides the driving force. Simple diffusion is one form of passive transport, but many substances cannot diffuse directly through the lipid bilayer, even when they are moving downhill.
They still require a membrane protein. When a molecule moves down its gradient with the assistance of a channel or a carrier, the process is called facilitated diffusion. The protein makes the route possible, but it does not make the transport active. This point is worth emphasizing because the presence of a transport protein does not automatically mean that ATP is being used. What determines whether transport is passive or active is the direction of movement relative to the gradient and the source of the driving force. Channels provide one way across the membrane. A channel is a selective, water-filled pore through which particular ions or molecules can pass. Channels are usually very fast because the transported substance does not need to bind and wait for the protein to move through a full conformational cycle. Once the channel is open, many particles can flow through in rapid succession. Channels are also selective. A sodium channel, for example, does not simply create an unrestricted hole in the membrane. Its structure favors sodium and excludes many other ions. Most channels are gated, which means they can open or close in response to a specific signal. Some respond to changes in membrane voltage. Others open when a ligand binds, and others respond to mechanical force. The gate determines when the route is available, but once the channel opens, movement through it remains passive. The substance travels down its gradient. Carriers work differently. A carrier binds a specific molecule on one side of the membrane, changes its shape, and then releases that molecule on the other side. Because binding and conformational change take time, carriers are generally slower than channels. They are also saturable. When every available carrier is occupied, increasing the concentration of the transported substance cannot increase the transport rate indefinitely. Carriers can participate in either passive or active transport. If the carrier moves its cargo down a gradient, the process is facilitated diffusion. If it moves the cargo against a gradient by coupling that movement to an energy source, the transport is active. So the distinction between channels and carriers is structural and mechanical. A channel forms a pore. A carrier binds its cargo and changes shape. But the distinction between passive and active transport is energetic. It asks whether the movement follows the gradient or opposes it. Water follows the same general logic. Water can move across the membrane directly to some extent, but in many cells, most water movement occurs through specialized channels called aquaporins. The passive movement of water across a selectively permeable membrane is called osmosis. Water moves toward the side with the greater concentration of osmotically active solute because that is the side where the effective concentration of free water is lower. The cell does not pump water directly during osmosis. Water moves in response to the solute difference that already exists across the membrane. This becomes extremely important in pathology because whenever the distribution of solutes changes, water follows. If sodium accumulates inside an injured cell, water enters with it, and the cell swells. We will return to that consequence when we study reversible cell injury. So, passive transport includes three major mechanisms. Simple diffusion allows suitable molecules to pass directly through the lipid bilayer. Facilitated diffusion allows substances to move down their gradients through channels or carriers. And osmosis describes the passive movement of water across the membrane, often through aquaporins. Active transport begins when the cell needs to move a substance in the opposite direction. Moving a substance from lower concentration to higher concentration is not spontaneous. It is the molecular equivalent of pushing something uphill. That movement requires energy. There are two main ways in which the cell pays for it. In primary active transport, the transport protein uses ATP directly. The transporter hydrolyzes ATP and uses the released energy to move ions against their gradients. These ATP-dependent transporters are the pumps, and they will be the subject of the next board. In secondary active transport, the transporter does not hydrolyze ATP directly. Instead, it uses the stored energy in the gradient of another ion. Usually, that driving ion is sodium. Sodium is allowed to move down the gradient that already exists across the plasma membrane, and its downhill movement is coupled to the uphill movement of another substance. The sodium-glucose cotransporter is a classic example. Glucose may need to enter a cell even when its intracellular concentration is already relatively high. Moving more glucose inward would therefore be energetically unfavorable. The transporter solves this problem by coupling glucose entry to the inward movement of sodium. Sodium moves downhill along its gradient, and the energy released by that movement carries glucose uphill against its own gradient. When the two substances move in the same direction, the transporter is called a symporter. When one substance moves in one direction while the other moves in the opposite direction, it is called an antiporter. Secondary active transport can therefore make it appear that the cell is moving one substance uphill without spending ATP, but the transport is not truly free. The sodium gradient being used as the energy source had to be created and maintained by an ATP-dependent pump. So the energy bill has not disappeared, it has only been paid earlier. This is the central logic of membrane transport. Moving down a gradient is energetically favorable. Moving against a gradient requires payment. Primary active transport pays directly with ATP. Secondary active transport spends a gradient that an ATP-dependent pump built beforehand. Every transport mechanism can therefore be understood by asking one question: who is paying for the movement? This question becomes especially important during cell injury. As long as the cell produces enough ATP, it can maintain ionic gradients and use those gradients to support secondary transport. But when ATP production fails, primary active pumps begin to stop. The gradients they maintained then begin to collapse. Passive downhill movement can continue because it does not require ATP, but the cell gradually loses its ability to push ions and molecules back uphill. Secondary active transport also fails because the gradients that powered it are no longer preserved. This is why energy failure produces such rapid consequences at the plasma membrane. The membrane itself may still be physically present, but the cell can no longer maintain the carefully controlled differences between inside and outside. Sodium moves into the cell. Potassium is lost. Water follows sodium. The cell begins to swell. The injury has not started because passive transport stopped. It has started because the energy-dependent processes that oppose passive movement can no longer keep up. A useful way to picture the system is to imagine water stored behind a dam. Passive transport is the water flowing downhill through an open route. The gradient itself drives the movement, so no additional fuel is required. Primary active transport is the pump that pushes water back uphill and refills the reservoir. That requires energy. Secondary active transport is a downhill stream turning a wheel that lifts a second load uphill. The second load appears to move without direct fuel, but the stream can turn the wheel only because someone paid to fill the reservoir in the first place. The one sentence to keep: small, nonpolar molecules can cross the lipid bilayer directly, while ions and most polar molecules require membrane proteins. Passive transport moves substances down their gradients without direct energy and includes simple diffusion, facilitated diffusion, and osmosis, whereas active transport moves substances against their gradients, either by using ATP directly or by using the stored energy of another ion gradient. Channels are fast, selective pores that mediate passive movement, while carriers bind their cargo and change shape, and may participate in either passive or active transport. We now understand the transport ledger, but we have not yet examined the machines that keep paying the bill. Which pumps maintain the ionic gradients of the cell, and what happens when ATP production begins to fail? That is where the next board begins. In the previous board, we said that every form of active transport eventually has to answer one question: where does the energy come from? Now we can look at the machines that pay the bill. There are many pumps in the cell, but two systems are especially important for understanding both normal cell physiology and the earliest stages of cell injury. The first is the sodium-potassium ATPase. The second is the group of calcium pumps that keep free calcium in the cytoplasm extraordinarily low. Both systems use ATP continuously, and both become dangerous the moment the cell can no longer supply that energy. Let us begin with the sodium-potassium pump. For every molecule of ATP it hydrolyzes, the sodium-potassium ATPase moves 3 sodium ions out of the cell and brings 2 potassium ions in. 3 sodium out, 2 potassium in. This may sound like a small exchange occurring across a tiny patch of membrane, but the pump performs several essential jobs at the same time. First, it establishes and maintains the characteristic ionic composition of the cell. Sodium is kept at a high concentration outside the cell and a relatively low concentration inside it. Potassium is arranged in the opposite way, with a high concentration inside the cell and a lower concentration outside. These gradients do not exist because sodium and potassium naturally prefer those locations. In fact, both ions are continually leaking down their gradients through channels and other pathways. The sodium-potassium pump must therefore run continuously to oppose that passive movement. It is not building the gradient once, it is rebuilding it every moment. Those ionic gradients are also fundamental to the resting membrane potential. The potassium gradient is especially important because potassium can leak out through resting potassium channels, leaving the interior of the cell relatively negative. The sodium-potassium pump maintains the sodium and potassium gradients that make this possible. Because it moves 3 positive charges out for every 2 it brings in, it also makes a small direct electrogenic contribution to the negative interior of the cell. So when we say that the pump helps establish the membrane potential, we mean both that it maintains the gradients on which the potential depends, and that its unequal exchange contributes directly to the charge difference. The second major job of the sodium-potassium pump is osmotic stability. This is where the pump becomes immediately important in pathology. Sodium is one of the major osmotically active ions in the extracellular fluid, and wherever sodium accumulates, water tends to follow. The cell contains proteins and other negatively charged molecules that cannot simply leave through the plasma membrane. These intracellular molecules attract positive ions and create a constant osmotic tendency for water to enter. If the cell allowed sodium to accumulate freely in the cytoplasm, water would follow it, the cell would expand, and its volume would become impossible to control. The sodium-potassium pump prevents this by continuously removing sodium from the cell.
So the pump is not simply maintaining an elegant biochemical gradient. It is one of the main reasons the cell does not fill with water. The third major job is to power secondary active transport. In the previous board, we used the sodium-glucose cotransporter as an example. That transporter does not hydrolyze ATP directly. Instead, it allows sodium to move back into the cell down its gradient, and it uses the energy of that movement to carry glucose uphill. But that system works only because the sodium concentration was kept low inside the cell in the first place, and the sodium-potassium pump is what created that condition. This means that the pump supports much more than sodium and potassium movement alone. Its gradient powers the uptake or exchange of many other substances. So one ATP-dependent pump supports membrane potential, cell volume, and a large network of secondary transport systems. That explains why the sodium-potassium ATPase consumes such a large share of the cell's energy. It is running constantly because the leaks are constant. Now, imagine what happens when ATP production begins to fall. The pump slows. Sodium is no longer removed as efficiently. But sodium channels and other passive routes do not suddenly disappear. Sodium continues to move inward down its gradient. Potassium continues to leak out. As sodium accumulates inside the cell, water follows. The cytoplasm expands. The endoplasmic reticulum begins to dilate. The cell becomes pale and swollen. This is cellular swelling, also called hydropic change, and it is the earliest visible manifestation of many forms of cell injury. The important point is that the membrane may still be physically intact. The cell has not necessarily ruptured. What it has lost first is the energy required to maintain the difference between inside and outside. The injury begins as a failure of control. If ATP production is restored before structural damage becomes severe, the pump can restart, sodium can be moved back out, water can leave, and the swelling can reverse. That is why cellular swelling belongs to the reversible phase of injury. The pump has stalled, but the system has not yet been destroyed. Now let us turn to calcium. Calcium presents the cell with a very different problem. The concentration of free calcium in the cytoplasm is kept approximately 10,000 times lower than the concentration outside the cell. The endoplasmic reticulum also serves as an intracellular calcium store, where calcium is held at a much higher concentration than in the surrounding cytosol. This means that the cell maintains an enormous calcium gradient across both the plasma membrane and the membrane of the endoplasmic reticulum. That low cytosolic concentration is maintained deliberately by ATP-dependent calcium pumps. One of these is PMCA, the plasma membrane calcium ATPase. PMCA removes calcium from the cytoplasm by pumping it across the plasma membrane and out of the cell. Another is SERCA, the sarcoendoplasmic reticulum calcium ATPase. SERCA removes calcium from the cytoplasm by pumping it into the endoplasmic reticulum, where it can be stored. Together, these pumps keep free cytosolic calcium at a very low resting level. And once again, they do not perform this task once and then stop. Calcium is constantly trying to move down its gradient into the cytoplasm. The pumps must continually oppose that movement. Why does the cell spend so much energy keeping cytosolic calcium so low? Because calcium is one of the cell's most useful signaling molecules. When the resting level is extremely low, even a small and temporary increase becomes easy to detect. A brief release of calcium can trigger contraction in a muscle cell. It can stimulate secretion. It can activate enzymes. It can alter metabolism.
and it can regulate gene expression. The signal works precisely because the baseline is so low. A small rise stands out clearly against the resting state. After the signal has completed its job, PMCA and SERCA remove calcium from the cytoplasm again, and the cell returns to baseline. So calcium signaling depends on a controlled cycle: release, response, and rapid removal. But the same design that makes calcium useful also makes it dangerous. The cell keeps a very large calcium gradient immediately across its membranes. As long as the pumps and membranes remain controlled, that gradient can be used in short, carefully measured pulses. If ATP production fails, however, the calcium pumps begin to stall. Calcium is no longer removed efficiently from the cytoplasm. It begins to enter from the extracellular space and to leak from intracellular stores. The controlled signal becomes a sustained rise. And calcium that is useful in a brief pulse becomes destructive when it remains elevated. High cytosolic calcium activates enzymes that can damage nearly every major component of the cell. It activates phospholipases, which break down membrane phospholipids. It activates proteases, which damage structural proteins and the cytoskeleton. It activates endonucleases, which fragment nuclear DNA. And it activates additional ATP-consuming enzymes at a moment when the cell is already running out of energy. Calcium can also worsen mitochondrial dysfunction, reducing ATP production even further and accelerating the progression toward irreversible injury. The complete destructive cascade becomes important when we reach the point of no return, but the principle we need here is simpler. The cell is not naturally at equilibrium with its environment. It stays alive by spending energy to resist equilibrium. Sodium is continually trying to enter, potassium is continually trying to leave, and calcium is held behind gradients so large that even a small loss of control can become dangerous. ATP-dependent pumps oppose those movements every moment. When ATP begins to fall, the plasma membrane does not immediately disappear. The energy-dependent systems fail first, while the physical boundary may still remain intact. The sodium gradient begins to collapse. Sodium accumulates inside the cell, and water follows. The result is cellular swelling, an early and potentially reversible sign that the cell can no longer maintain its internal environment. At the same time, calcium is no longer removed efficiently from the cytoplasm. What should have been a brief signal becomes a sustained rise, activating enzymes that damage membranes, proteins, DNA, and mitochondria. The two failures, therefore, begin with the same unpaid energy bill, but they produce different consequences. For sodium, think of a bilge pump working continuously against a slow leak. As long as the pump has power, the interior remains dry. When the power fails, the water does not need to be invited in. It simply follows the path that was already available. Calcium is better pictured differently. It is a weapon kept behind a locked door. In small, controlled releases, it carries information. But when the pumps lose power and the lock fails, the same stored gradient becomes destructive. The one sentence to keep: every gradient the cell maintains is a debt paid in ATP. The sodium pump keeps the cell from flooding. The calcium pumps keep a weapon locked outside the cytoplasm. And when the payment stops, the water walks in and the weapon is released. So far, every substance we have moved across the plasma membrane has crossed one molecule or one ion at a time. Some passed directly through the lipid bilayer, while others used channels, carriers, or ATP-dependent pumps. But none of those mechanisms can solve the problem of size. A whole protein cannot pass through an ion channel. A bacterium cannot bind to a carrier and be transported across. A dead cell is far too large for any molecular pore. To move cargo of that size, the membrane has to stop behaving like a flat border and begin changing its shape. It bends around the material, encloses it, and pinches off a membrane-bound compartment called a vesicle. This is bulk transport. When the plasma membrane bends inward and brings material into the cell, the process is called endocytosis.
When an intracellular vesicle fuses with the plasma membrane and releases its contents outside, the process is called exocytosis. These two processes move material in opposite directions, but they depend on the same fundamental property. The membrane is fluid enough to bend, fuse, separate, and rebuild itself without losing the boundary of the cell. Let us begin with endocytosis. Endocytosis is not one single mechanism. The cell uses different forms depending on what it needs to bring in, how large the material is, and how selectively that material must be recognized. The most selective form is receptor-mediated endocytosis. Here, the cell does not simply engulf whatever happens to be nearby. A specific extracellular molecule first binds to a specific receptor on the plasma membrane. The receptor cargo complexes then gather into specialized regions known as clathrin-coated pits. Clathrin proteins assemble along the cytoplasmic surface and help bend the membrane inward. As the pit deepens, a protein called dynamin forms a ring around the narrow neck of the budding vesicle and helps pinch it free from the plasma membrane. The result is a clathrin-coated vesicle carrying a selected cargo into the cell. The clathrin coat is then removed, and the vesicle delivers its contents to an early endosome. The early endosome is not simply a waiting room. It is a sorting station. Its interior becomes acidic, and that fall in pH causes many receptors to release their cargo. The receptor can then be packaged into a recycling vesicle and returned to the plasma membrane, ready to be used again. The cargo, meanwhile, may continue deeper into the endosomal system and eventually reach a lysosome for processing. This recycling step is what makes receptor-mediated endocytosis economical. The cell does not need to manufacture a new receptor for every molecule it internalizes. The same receptor can bind cargo, enter the cell, release that cargo in the acidic endosome, return to the surface, and begin the cycle again. The classic example is the uptake of LDL. LDL particles carry cholesterol through the blood, but the particle is too large and too polar to cross the lipid bilayer directly. Instead, LDL binds to the LDL receptor on the cell surface. The receptor cargo complexes cluster in clathrin-coated pits. Dynamin helps release the vesicle, and the LDL is delivered to the early endosome. As the pH falls, the LDL separates from its receptor. The receptor returns to the plasma membrane, while the LDL particle is directed toward lysosomes, where its cholesterol can be released for use by the cell. This is a remarkably efficient system, but it also gives us one of the cleanest examples of how a normal membrane mechanism becomes disease. In familial hypercholesterolemia, the LDL receptor pathway is defective. The receptor may be absent, unable to bind LDL, unable to cluster correctly in coated pits, or unable to recycle normally, depending on the underlying mutation. Whatever the precise defect, the result is the same. Cells cannot remove LDL from the blood efficiently. The LDL remains in the circulation, plasma cholesterol rises, and cholesterol accumulates in arterial walls, producing premature atherosclerosis. The disease is therefore not caused because LDL has suddenly become impossible to carry.
It is caused because the membrane can no longer recognize and internalize it correctly. A normal transport mechanism has failed at the level of the receptor. Clathrin-coated pits are not the only specialized membrane invaginations used for endocytosis. Some cells also contain caveolae, small flask-shaped invaginations rich in cholesterol, sphingolipids, and proteins called caveolins. Caveolae participate in selected forms of endocytosis, signaling, and transcytosis, the movement of material across a cell from one surface to another. They are particularly prominent in cells such as endothelial cells, where substances may need to cross the entire cellular barrier without being released into the cytoplasm.
So even within endocytosis, the membrane uses different entry routes for different jobs. Pinocytosis is less selective. The name literally means cell drinking. Instead of recognizing one specific particle through a dedicated receptor, the cell takes in small amounts of extracellular fluid, together with whatever dissolved molecules happen to be present. The membrane forms small vesicles and samples the environment around the cell. Pinocytosis therefore provides fluid phase uptake rather than the highly selective capture of one particular ligand. Phagocytosis operates on a much larger scale. The name means cell eating, and that description is quite accurate.
During phagocytosis, the cell extends its plasma membrane around a large particle, such as a bacterium, a fragment of dead tissue, or an entire dying cell. But before the cell can engulf the target, it usually has to recognize it. Sometimes the target carries molecular patterns that can be detected directly by receptors on the phagocyte. In many cases, however, recognition becomes much more efficient after the target has been coated with molecules called opsonins. Immunoglobulin G can coat the surface of a microbe and bind to Fc receptors on the phagocyte. The complement fragment C3b can coat the same target and bind to complement receptors. The opsonin marks the particle, and the receptor gives the phagocyte something to grip. The details of those immune systems belong to the immune pathway, but the membrane logic is already visible here. Recognition comes first, and engulfment follows. Once the target is recognized, the actin cytoskeleton pushes the membrane outward to form pseudopods. These extensions gradually surround the particle until their edges meet and close, trapping the target inside a large intracellular vesicle called a phagosome. The phagosome alone does not necessarily destroy what it contains. To become a destructive compartment, it must mature and fuse with lysosomes. The fused structure is called a phagolysosome. Inside the phagolysosome, the target is exposed to an acidic environment, lysosomal enzymes, reactive oxygen species, and other antimicrobial mechanisms. Recognition identifies the target. The membrane encloses it, and the phagolysosome performs the killing and digestion. This sequence is central to inflammation. Neutrophils reaching a site of acute infection use it to engulf and destroy microorganisms.
Macrophages use it to ingest microbes, damage tissue, foreign particles, and cellular debris. But once again, the same protective mechanism becomes a site of disease when one of its steps fails. Mycobacterium tuberculosis can survive inside macrophages, partly because it interferes with the normal maturation of the phagosome and its fusion with lysosomal compartments. The organism has been engulfed, but engulfment alone is not enough. If the phagosome does not become a fully functional phagolysosome, the microbe can remain alive inside the very cell that was supposed to destroy it. That failure helps explain persistent intracellular infection, chronic inflammation, and the eventual formation of granulomas. A related lesson appears in Chediak-Higashi syndrome. Here, a defect in lysosomal trafficking impairs the normal movement and fusion of lysosomal granules. Phagocytes may recognize and engulf microorganisms, yet their ability to deliver the full lysosomal killing machinery becomes defective. The failure is not at the level of finding the target, it is at the level of bringing the destructive compartments together correctly. Phagocytosis can also damage tissue even when the phagocyte itself is functioning normally. Sometimes the target is simply too large or too firmly attached to be completely enclosed. A macrophage may attempt to engulf a large crystal, a long silica particle, or an immune complex deposited along a basement membrane, but it cannot close its membrane around the target. This is called frustrated phagocytosis. The phagocyte has activated its destructive machinery, but because the target cannot be sealed inside a phagolysosome, enzymes and reactive molecules are released into the extracellular space instead. The machinery intended to destroy the target now damages the surrounding tissue. This mechanism helps connect phagocytosis to diseases such as silicosis, crystal-induced inflammation, and immune-mediated injury along tissue surfaces. The defense response has become the lesion. Phagocytosis is also used for something quieter than antimicrobial defense. It removes cells that have completed apoptosis. In the first board of this video, we saw that a dying cell exposes phosphatidylserine, or PS, on the outer surface of its plasma membrane. That exposed PS acts as an "eat me" signal. Macrophages and neighboring phagocytes recognize the signal, surround the apoptotic cell, and remove it before its membrane ruptures. This specialized clearance of apoptotic cells is called efferocytosis, and this closes a loop that began at the start of the membrane story. A change in the membrane of the dying cell marks it for removal, and the membrane of the phagocyte then wraps around it and carries it away. Because the apoptotic cell is engulfed while its contents remain contained, intracellular material does not spill freely into the surrounding tissue. Death remains quiet. When efferocytosis fails or is delayed, however, the apoptotic cell may progress to secondary necrosis. Its membrane loses integrity, its contents escape, and a death that should have remained contained can become inflammatory. So the difference between silent death and inflammatory death depends partly on whether bulk transport happens in time. Now, consider the opposite direction. Cells do not only bring material inward, they also need to send material out. That is the role of exocytosis. In exocytosis, an intracellular vesicle moves toward the plasma membrane, docks with it, and then fuses with it. Once the two membranes merge, the vesicular contents are released into the extracellular space. This is how endocrine cells secrete hormones, how neurons release neurotransmitters, and how glandular cells release enzymes and other proteins. Some exocytosis occurs continuously as part of the ordinary maintenance of the cell surface. Other forms are tightly regulated and occur only when the cell receives a specific signal. Calcium is often the final trigger. A brief rise in cytosolic calcium can cause a secretory vesicle to fuse with the plasma membrane and release its contents almost immediately. This gives us the other face of the calcium story from the previous board. When the increase is brief, localized, and controlled, calcium carries information and triggers useful events such as neurotransmitter release. When the increase becomes sustained, because the pumps and membranes have failed, the same ion activates phospholipases, proteases, endonucleases, and other destructive pathways. The difference is not simply the molecule, it is the amplitude
location, and duration of the signal. Pathology begins when the cargo, the signal, the quantity, or the timing becomes abnormal. Exocytosis also does more than release cargo. Every time a vesicle fuses with the plasma membrane, the vesicle membrane itself becomes part of the cell surface. Exocytosis therefore adds lipids and proteins to the plasma membrane. Endocytosis does the opposite. When a vesicle pinches inward, part of the surface membrane is removed. The cell must balance the two processes so that secretion and uptake do not continuously enlarge or shrink the plasma membrane. The border is maintained through continuous renewal. Parts of the membrane are constantly internalized, sorted, recycled, degraded, replaced, and returned. The plasma membrane is not a permanent sheet whose molecules remain fixed for the life of the cell. It is a maintained pattern. Now we can return to the basic problem that bulk transport solves. The bilayer protects the cell precisely because it blocks many things. But that same barrier would also prevent the cell from obtaining selected macromolecules, clearing debris, defending itself against microorganisms, or releasing the products it has made. Bulk transport allows the cell to cross its own border without opening a permanent hole through it. The membrane wraps around selected material, carries it inside a sealed vesicle, or fuses with a vesicle to release material outside, while preserving the distinction between the cytoplasm and the extracellular environment. The normal mechanisms we have described plant several major pathology seeds. A defective LDL receptor produces familial hypercholesterolemia because receptor-mediated uptake fails. Failure of phagolysosome formation allows intracellular microbes to survive. Frustrated phagocytosis turns a protective response into collateral tissue injury. Failed epherocytosis converts quiet apoptotic death into inflammation. And uncontrolled exocytosis, secretion, or signaling can become pathological when the cargo, quantity, or timing is wrong. A useful way to picture the plasma membrane is not simply as a wall, but as a pair of hands. Those hands selectively collect LDL and other ligands, drink from the surrounding fluid, grip opsonized microbes, swallow foreign particles, and bury the dead. They turn outward to deliver hormones, enzymes, and neurotransmitters. But the hands can also fail to recognize their cargo, fail to digest what they have swallowed, or release their destructive contents in the wrong place. The same membrane machinery that protects the cell can therefore become the mechanism of disease. The one sentence to keep: the membrane is not only a wall, it is a pair of hands. Endocytosis selects what enters. Phagocytosis swallows microbes and the dead. And exocytosis releases what the cell must send out. We began this video with a membrane that created an inside and an outside. We then saw that the barrier is selective, that crossing it follows an energy ledger, and that ATP-dependent pumps keep the cell away from the equilibrium that would destroy it. Finally, we saw that when cargo becomes too large to cross through a protein, the membrane itself bends, extends, pinches, fuses, and moves around it. But the membrane cannot generate those movements by itself. A phagocyte needs force to extend pseudopods around a microbe. A vesicle needs tracks and motors to travel through the cytoplasm. A receptor must be carried back to the surface after it has released its cargo. And a dividing cell must reorganize its entire structure from within. That force, those tracks, and that internal organization come from the cytoskeleton.